Good morning, everyone. Today's topic is about erythropoiesis. Erythropoiesis, first we'll see about the definition. It is the process of origin, development, and maturation of erythrocytes or the red blood cells. Hematopoiesis is the process of origin, development, and maturation of all the blood cells. Erythropoiesis is just origin and development and maturation of erythrocytes. So next we'll see about the site of erythropoiesis in fetal life as well as children and adults. So first, in fetal life, the erythropoiesis occurs in 3 stages. So the first stage is mesoblastic stage. It occurs during the first 2 months of intrauterine life. RBCs are produced from the mesenchyme of the yolk sac. The second stage is hepatic stage from the 3rd month of intrauterine life. Here the liver plays a main role for the production of RBC cells, and the spleen and lymphoid organs are also involved in the formation of erythrocytes. The next one is myeloid stage. So the stage occurs during the last 3 months of intrauterine life. In newborns, children and adults, so up to the age of 20 years, the RBC cells are produced only from the red bone marrow of the long bones as well as the flat bones. But after the age of 20, RBC cells are produced from the membranous bones, for example, vertebrae, sternum, ribs, scapula, etc. So after 20 years, the shaft of the long bones become yellow bone marrow because of the increased deposition of fat, and it loses the erythropoietic function. So in adults, liver and spleen plays a major role for the blood cell formation. So 75% of bone marrow is involved in the production of leukocytes, and only 25% is involved in the production of RBC cells. But still, leukocytes are less in number than the erythrocytes. So next we will see about the changes during erythropoiesis. So the cells from colony forming unit E passes through different stages and finally become the matured RBC cells. So the changes that occurring during these processes are: one is reduction in the size of the cell, that is from the diameter of 25 to 7.2 micron, which is the normal diameter of the red blood cell. And second changes that are occurring during these processes are disappearance of nucleoli and the nucleus. And the third change is appearance of hemoglobin. And the last one is change in the staining properties of the cytoplasm. So the stages occurring between these colony forming and the matured RBC cells are: one is pro-erythroblast, second early normoblast, third intermediate normoblast, fourth one is late normoblast, fifth one is reticulocyte, and finally it becomes a matured erythrocyte. So let us learn about one by one. So the first stage is pro-erythroblast. It is also known as megaloblast. So the pro-erythroblast or megaloblast is the first cell derived from the colony forming unit E. So it is very large in size with a diameter of about 20 micron. So it contains nucleus and it is very large. It occupies the entire cells. The nucleus has 2 or more nucleoli and a reticular network. The pro-erythroblast, it does not contain any hemoglobin. It multiplies several times and finally forms the next stage called early normoblast. So the early normoblast is little bit smaller than the pro-erythroblast with a diameter of about 15 micron. So during pro-erythroblast, its size is about 20 micron. Now it becomes 15 micron. And in the nucleus, now the nucleoli disappears. So the condensation of chromatin network occurs in this early normoblast stage. So this cells develops into the next stage called intermediate normoblast. So during intermediate normoblast, the cell even becomes smaller than the early normoblast, and its diameter is about only 10 to 12 micron. But still the nucleus is present in this stage. Here the new hemoglobin starts to appear. So this cell develops into a next stage called late normoblast. So let us learn about the late normoblast. So the late normoblast, the diameter of the cell decreases further to about 8 to 10 micron. Now the nucleus becomes very small and the quantity of the hemoglobin increases. In the final stage of the late normoblast, the nucleus disintegrates and disappears in this stage. So the next stage is reticulocyte, which is also known as immature RBCs, and it is slightly larger than the matured cell. So the cytoplasm contains the reticular network, and the cell is in this stage it is known as reticulocyte. Here the number of reticulocytes decreases during the first week after birth. During this stage, the cells enters easily through the blood capillaries from the site of production by the process known as diapedesis. So the last final stage is matured erythrocyte or matured RBC cell. Here the reticular network starts to disappear and the cell becomes completely matured and attains the normal shape, which is biconcave in shape, and the cell decreases in size to about 7.2 micron. And the matured RBC is with hemoglobin but without nucleus. So it requires about 7 days for the complete development and maturation of RBC cells. So the up to reticulocyte it takes for 5 days, and the last two stages 2 days. And the duration, it requires 5 days up to the stage of reticulocyte, and the reticulocyte takes 2 more days to become the matured erythrocyte or the RBC red blood cells. Now we will see about the factors necessary for this erythropoiesis. So formation of erythropoiesis is occurring by these factors. First one is general factors, which includes the erythropoietin hormone, thyroxine hormone, hematopoietic growth factors, and the vitamins. Second one is maturation factors, which includes the vitamin B12 and the folic acid. And the last one is factors necessary for the hemoglobin formation or proteins and amino acids, minerals like iron, copper, cobalt, nickel, etc., and the vitamins. So that's all about the erythropoiesis, how the erythrocytes are formed. Thank you.